Okay, for our final talk this morning, we have Dr. Paolo Vineis and causal models in epidemiology.
Thank you for inviting me to this workshop. In fact, I will talk mainly about the concept of the exposome and also some specific results we have obtained from our research, which is called Exposomics. How can I go on? Right. Okay, so I have three parts in my presentation. One is about how we conceive and interpret carcinogenesis based on recent advancements. Then there is a conceptual part concerning the exposome, and then examples, as I said, from Exposomics research. So first of all, well, we know that cancer is a multistage and multifactorial disease, which means many different things. For example, it means that in any population you have a small number of people who lacks just the activation of the last stage or hit to complete the carcinogenic process. Then you will have a larger number of individuals who lack the activation of 2 stages, 3 stages, and so on, and probably a small number of people who have no activated stage. The problem is that we do not know who those people are. We are not able yet to identify those people, with exceptions. An obvious exception is genetic susceptibility based on familiar conditions, for example. This is a very popular way to talk about causation in epidemiology, which has been put forward by Ken Rothman many years ago, and is still valid. It is called the sufficient component causal model, but also Rothman's pies. What does it mean? Each of those pies refers to a single individual. Each of them is a sufficient complex of exposures. None of the exposures or hits is sufficient itself. Usually for non-communicable diseases, none of them is necessary, though we know examples of necessary causes. Probably HPV is necessary for cervical cancer. So this is a very useful way to describe causation in epidemiology, but it has at least one problem. That is, it doesn't take time into account. So there is also a temporal component, which is very well expressed by the evolutionary interpretation of cancer. Cancer is now interpreted as a Darwinian process in the single individual. Essentially, cancer phenotypic changes branch since early stages, and the phenotype becomes more and more complex and even individualized. So each individual has her own history of carcinogenesis due to the history of branches and branching that occurred in time, so that you have different phenotypes or different mutations, even in the single tumor or in metastasis. So this temporal component is not really present in Rothman's model, and it's not present also in another model which has been put forward recently, well, not so recently, which is the hallmarks of cancer by Anahan and Weinberg. So we have to consider the temporal component because it fits very well with the Darwinian interpretation. I cite this paper by Gerlinger, which is very clear about the idea of branching evolution. The selective advantage of any genotype is dependent on the environment. So you do not only have mutations, but you have a history of mutations, and also the interplay between mutations and the environment. The environment must be defined broadly to include the cell's repertoire of gene and protein expression, the mutations and epigenetic changes already present in that cell, the local external microenvironment, distantly acting factors such as hormones and factors external to the host like carcinogens and exogenous therapies. Some people have coined the word selectogens. So in addition to mutagens, you also have environmental selectogens, meaning exposures that select for certain advantageous mutations. And the final product is a combination of both. Not only mutations. Well, obviously, there are some driver mutations where environmental variation may matter less because such mutations almost invariably confer a selective advantage, but this is the exception more than the rule. So this is a representation of the Darwinian evolution of cancer, where you have a number of mutations, but also bottlenecks, selective pressures coming from non-mutagenic environments. It is likely that some of the exposures we refer to in epidemiology are not mutagenic. We can think of obesity, dietary components, and so on, but they act by selecting existing mutations. And also the relevance of a certain mutation depends on the previous mutations in the same cell and the following subsequent mutations. So it is a historical development. Now, in epidemiology, we use biomarkers. And in fact, what I'm trying to do is to see how we can combine epidemiological tools which refer to external exposures with mechanisms. And biomarkers are very much used to make inferences about mechanisms and also to link exposures with the mechanisms. So one assumption is that molecular epidemiology is not really interested in biomarkers per se, unlike clinical research, but it is more interested in the continuum of disease development. So it is interested in processes rather than in things. Biomarkers are included in epidemiology and molecular epidemiology in particular because we are interested in processes that lead from exposure, exposure biomarkers, early response, and the final outcome. Well, I refer to this chapter in a book, which is now becoming a paper, because we try to expand these ideas there with a philosopher. Now, there are several approaches to causality, obviously in philosophy, but also philosophy applied to biological sciences. And I like referring to Wesley Salmon, who posed the question, how can we distinguish between causal processes, because we are talking about processes, rather than ontology, what biomarkers really are. We are looking at biomarkers as processes, or as particular images of the process. How can we distinguish genuine causal processes from non-causal processes? And what he proposed was that processes are word line of objects. For example, airplanes fly in the sky, express word lines, but so the same is true for shadows on the ground. How can we discriminate between word lines or processes that are causal and those that are not? In Salmon's approach, causal processes are those which are capable of transmitting conserved quantities, such as mass, energy, momentum, or charge, whereas shadows do not transmit conserved quantities. When the airplanes collide, there is a transmission of conserved quantities, so that they interfere with each other physically, whereas the shadows on the ground do not transmit conserved quantities. So the latter do not represent a causal process. But in fact, the transmission of conserved quantities is a useful concept in physics, not necessarily in, or at least in a different way in biology and in medicine. And our proposal in the chapter I mentioned is that transmission of conserved quantities is also transmission of information via molecules. So probably transmission of information via molecules is what we call the exposome. It is the transmission of information, in fact, from outside the body to inside the body through changes in molecules and through a chain of changes that goes from the genome to the transcriptome to the proteome to the metabolome. And changes in the genome could be inherited, highly penetrant variants, they can be mutations, they can be epigenetic changes, and so on. So we believe that the concept of transmission of information is key to understand how to connect external exposures with the internal exposome. One advantage of this idea is that it is potentially widely applicable and capable of explaining also complex factors like social factors. I think that John Samet also mentioned social factors, how society influences disease. And so far, there has been a disconnect between social interpretations, social sciences, biological sciences, and so on in relation to disease. But it is possible that using this paradigm of information transmission and also exposome research, we are able to connect micro and macro mechanisms and social determinants. Which is an example of, well, an example of this is a consortium funded by the European Commission, which is called LifePath in Europe, that I coordinate, where we want to look at different layers, including life course socioeconomic status, intermediate factors like environmental exposures, psychosocial exposures, and behavioral exposures, and then the mechanisms that lead to, in our case, healthy or unhealthy aging. And there is a number of potential mechanisms in the middle between those exposures and the overarching concept of life course, socioeconomic status, and healthy aging. So we want to use a combined approach which uses the traditional epidemiological approaches based on questionnaires and so on, but also mechanistic information on the intermediate pathways. But I would like to refer mainly to another European Commission-funded consortium, which is called Exposomics. Well, the original idea, this is a citation from the call. So the aim was to develop a novel approach to the assessment of exposure to high priority environmental pollutants, and we chose air pollution and water contamination, by characterizing the external and the internal components of the exposome. Well, you may know that the idea of exposome was coined by Chris Wilde many years ago, and the idea was coined essentially to draw our attention to the fact that a lot of efforts have been put into genomics, whereas not much has been done to improve the science of exposure. We know that exposure measurement is affected by a large measurement error, whereas measurement error in genomics is much smaller. And also another big problem in environmental epidemiology is intra-individual variability. Exposures change all the time, possibly second by second, whereas the genome is, at least the sequence of DNA bases is stable in the course of life. So according to Chris Wilde, the concept of the exposome refers to the totality of environmental exposures from conception onwards and includes both external and internal components. And there is also a very important temporal component. As I said before, we have to consider events in a sequence and the history of these events, whether they are external exposures or internal changes. Then the concept of the exposome has been refined by Martin Smith and Steve Rappaport in a seminal paper. In our case, we decided to focus on air pollution and water contamination during critical periods of life, because the life course approach is also crucial in the exposome concept. So our project, Exposomics, centers on one side on advanced exposure assessment at the personal and population levels within existing European cohorts. And second, using multiple omic technologies for the analysis of biological samples. Now, what I want to show is how these approaches can possibly contribute to causal reasoning. Based on what I said before about Salmon's model and the idea of transmission of information. Well, Exposomics' work is in fact quite relevant to this 21st century hazard identification and risk assessment report described by John Sammet, where central is the paradigm of pathway perturbation. In the case of Exposomics, we have identified 7 areas where exposome research can be fruitful, again referring to air pollution and water contaminants. One is exactly pathway perturbation and causal assessment in a way that we call meet in the middle. I will explain what it means. Then also mixtures, which is still a kind of uncertain area. We don't have really the tools to disentangle the impact of single components in mixtures, or to see whether, to infer whether the mixture has a health impact which is greater than the sum of its components. Then dose response, particularly at low levels of exposure, is another open field, very often because of low power of epidemiological studies. Crossomics, I will explain later what it is. It is the joint analysis of different omic results. Then calibration of health effects is a potentially nice contribution of exposome work to decrease misclassification and increase the power of our studies. Then we use longitudinal models, and we have statistical tools to look at the temporal sequence of exposures. And then the concept of data integration is clearly crucial. Well, pathway perturbation is key, as we have heard already. And in several ways, exposome research shows that omics and molecular work can identify early signs of damage from environmental agents and could be used for prediction. Or at least this is the challenge, and we have early results. The problem is that obviously pathways are complex. So there is not a one-to-one relationship, not only between exposure and disease, but also between exposure, intermediate changes, molecular changes or pathways, and the outcomes. So that different chemicals can act through different pathways, and these pathways can lead to different diseases, which complicates the picture, but it is probably a reality, and we have to take that into account. Sorry. So one example of our research, this is metabolomics applied to contamination of water by chlorination by products. Well, you may know that chlorination by products or disinfection by products have been suspected of leading to an increased risk of bladder cancer in particular. Some molecular work has been done already, like genotoxicity, micronuclei in swimmers. There is a series of studies done in swimmers, particularly in Barcelona. So this is one of those studies, which is part of Exposomics. We enrolled 60 volunteers who swam for 40 minutes. We measured disinfection by products in the water and in exhaled air. And we took blood. And in that blood, we measure 5 different omics, which are proteomics, transcriptomics, metabolomics, epigenomics, and adductomics. And I will explain later what adductomics is. So this graph shows that there is indeed quite a different pattern of metabolomic features in the blood after swimming and before swimming. After swimming is in blue, and before swimming is in orange. Certainly swimming modifies the metabolomic pattern in people. This is in blood. However, causal inference is not straightforward, because there is an interference from physical activity. So this figure doesn't lead us to conclude that the 2 metabolomic patterns are due to disinfection by products. There is a physical activity component. And we are now working to disentangle the impact of physical activity from the impact of disinfection by products. And in fact, we have found 25 different metabolomic features that refer to chlorinated compounds. So they are attributable to swimming rather, to being exposed to disinfection by products rather than to physical activity. But this is just to give an idea of what you can get from one particular omic to study the intermediate pathways, but also the questions that this raises about potential confounders. This is a pre-post study. Another concept we are developing in Exposomics is meet in the middle. What does it mean? Well, so far, for most exposures, as far as I know, people have looked at the association between intermediate markers and the outcome, and then separately, literally in other studies, in other populations, at the association between intermediate markers and exposures. But very rarely people have done they have looked at the whole picture in the same population longitudinally. That is, they looked at exposure, then intermediate markers, and the relationship between intermediate markers and the outcome in the same groups of people. This is because you need cohorts in which you look at the perturbation of intermediate markers in relation to previous exposures, so you need historical exposure assessment, and then prospectively in relation to the onset of disease. This requires long-term longitudinal studies with a sufficiently long follow-up. Now, we have some proof of principle that meet in the middle approach may work. And in fact, we have a paper coming from Exposomics, which is about cardiovascular diseases. And what we show is that not only air pollution is related to cardiovascular diseases, which has been already shown by others. In fact, there are several studies showing that. But we also observe that air pollution is associated with some oxidative stress and inflammatory markers. And in turn, the same markers are associated with the risk of cardiovascular disease. So in the literature, you will find the first association, and you will find the second association, but you will not have the full picture. And by the way, both branches have been done agnostically. I mean, we looked at the association with disease first, and we selected signals. We looked at air pollution second, and we selected the significant signals, and the signals were the same. So this is quite interesting evidence that the meet-in-the-middle approach can reinforce in some way causal inference by including mechanistic reasoning within the black box. So we are opening the black box. This is another example of an omic approach. This is genomics. Usually when people talk about genomics, they think of inherited gene variants or mutations. But this is somatic mutations. So these are acquired mutations. This is a study done by Mike Stratton in Cambridge, in which we participated. And he looked, essentially he did a whole genome sequencing of biopsies from different types of cancer and comparing smokers and non-smokers. And what came out was that signatures, mutational signatures, were quite different for certain cancers compared to other cancers. For example, there is a signature 4, which is characteristic of lung cancer and larynx cancer and upper aerodigestive tract cancers, suggesting that this is characteristic of the sites that, or organs that are directly affected by smoking, have a direct contact with smoke, whereas other signatures are more typical of bladder cancer or pancreatic cancer. So first, this allows us to infer that there is a degree of specificity of mutational signatures depending on the cancer site. But also, this is quite interesting, in cells in culture treated with benzopyrene, Mike Stratton found the same signature as found, or a very similar signature as found in biopsies from smokers with lung cancer, which may lead to say something, to make an inference about the relevant chemicals in tobacco smoke that lead to lung cancer, as opposed to bladder cancer, where it is more likely that the chemicals involved are aromatic amines. So this is interesting in the context of mixtures. Tobacco smoke is a very complex mixture. And so this kind of omic work, which is somatic genomics, can help disentangling the different, the impact of the different components in mixtures. Then another unresolved issue in epidemiology is dose response at low levels of exposure, because very often we don't have enough power, particularly for rare outcomes. Now, one of the assumptions of exposome work is that omic changes are more frequent than the final outcomes, like single cancers. So you can look at perturbation of pathways or omic features with greater statistical power compared to epidemiological studies with outcomes like cancer. Well, this is still preliminary, but it is the kind of work we are doing in Exposomics because we have cohorts with huge differences in levels of exposure. This is a PM10, and this is a component of Exposomics in children. And we have children coming from Turin, where I come from, by the way, exposed to very high levels of PM10 or PM2.5, whereas children from Belgium, the environ age cohort, have much lower levels of exposure. And this slide just shows that we are able to identify statistically significant metabolomic signals in Turin and not in environ age in Belgium, well, except, no, none of them. Well, I don't want to go too far in the interpretation of that, but it might not be by chance that you see many more signals, some of which are statistically significant after Bonferroni correction in Torino as opposed to Belgium, as opposed to a low exposure area, which is in fact partly a rural area. So we are working along these lines to try to connect the levels of exposure with omic signals. And it is possible that we can show that you have indeed early omic perturbations also at low levels of exposure that can be found many years before the onset of disease. Then in Exposomics we apply this concept of regression calibration. What does it mean? This is also part of the exposome concept because, as I said before, the whole idea of the exposome was suggested by Chris Wilde because of the limitations of current exposure assessment with the tools that epidemiology now has. So there is a whole branch of the exposome field which aims at developing new tools like sensors for personal exposure monitoring. And the conundrum there is that you cannot apply these sensors, which tend to be expensive, to large populations, to whole cohorts. So one way to overcome this limitation is to improve exposure assessment in a subset and then use these measurements for calibration of the association estimates in the larger cohorts. For example, in this case, we collected personal exposure monitoring data on PM2.5, but also ultrafine particles with backpacks used by 42 individuals in Turin, but overall they were 200 in Exposomics. We had 3 measures per individual in the course of 1 year. So we have what we call true exposure, which is probably an overstatement, but it is based on personal backpack measurements. Whereas a usual surrogate exposure is based on outdoor measurements, like monitoring stations. So there is a statistical approach called regression calibration, where you can adjust for the attenuation or measurement error due to the surrogate exposure measurement, which is the one usually included in epidemiology, and see whether the association with disease becomes stronger. And this is what we find in relation to cardiovascular diseases. The dotted line is without calibration. So I believe that the odds ratio was 1.2, but the red line is after calibration, and the relative risk becomes much higher. So this is a way to overcome the problem that you cannot use sensors and maybe even omic measurements in large cohorts, thousands or tens of thousands of people. But you can calibrate risk using this methodology, which was put forward by Spiegelman years ago. Then, as I said, one of the assumptions of the exposome is that you have to take into account potentially the whole life of exposures, the whole experience of exposure across life of an individual, because the outcome is a historical event. And so we are trying to look at the longitudinal behavior of omics, omic measurements. This is DNA methylation. As I said, in Exposomics we have 5 different types of omics measured in all individuals. We have these measurements for between 2,000 and 3,000 people, the 5 I mentioned. So this is epigenetics, and it is usually Lumina for 50K methylome. And we are using a particular cohort in England, which is called ALSPAC, which has the characteristic of having collected blood and exposure estimates at different times in newborns at the age of 5, 7, and then again, I believe, 10. So we have blood for all of those time points, and we are looking for example, signals that are constant across the different time points. And we find one. We don't know what it really means. This MED1, MED1L1 gene. Well, once again, this is, so to say, proof of principle, development of tools, and so on. But the idea is that you can look at signals that remain across different time periods, and that can say something about a certain type of pathway perturbation, or signals that are more typical of sensitivity windows. Okay, of the omic technologies I mentioned, there is one which is new, which is adductomics. And this has been developed by Steve Rappaport first, and then transferred to the King's College laboratory led by David Phillips. So now we have as many as 2,000 adductomic measurements in Exposomics. And this is based on albumin adducts. And the assumption is that you may, since this is an agnostic search, by definition, because omics are all agnostic, they look at the whole set of a certain kind of molecule. They can allow you to identify, for example, new chemicals that are in the air adducted to albumin. So far, we have measured 2,000 samples in adductomics by David Phillips. We have also parallel studies which are smaller in Steve Rappaport's lab. We did some quality checks, and the correspondence is fair but not complete. So we are also, say, improving the quality and validating the method. And well, the advantage of this compared to metabolomics, for example, is that the half-life is longer compared to metabolomic features in the blood. Okay, very quickly, I would like to say something about precision prevention, because sometimes these technologies and the input of molecular epidemiology into molecular tools into epidemiology is focused on potential uses according to the precision medicine or precision prevention paradigm. And I would like to say a few words of caution, because there are several limitations in the precision prevention paradigm, and with Chris Wilde we have written a short piece in the Lancet Oncology which is in press. Well, first of all, we believe that there is some confusion between precision applied to individuals as opposed to precision applied to molecules. So whatever you find with molecules not necessarily is able to predict what will happen in individuals. So precision at the molecular level may give the false impression of a curative or preventive power that is absent when transferred into practice. We have a lot of examples from clinical research. And so we have to be cautious. We still need epidemiological, probably classical epidemiological approaches to show that what is true for molecules is also true for humans. Second, well, it is very likely that there are multiple pathways involved in a certain relationship between exposure and disease, but also treatment and disease. One of the best examples of precision prevention nowadays is the selection of individuals with variants in the prostaglandin pathway that modify the response to aspirin chemoprevention to prevent colorectal neoplasia. However, there are multiple pathways there, not a single pathway, which means that we need to identify individuals who do not have a single characteristic. That would be very easy, but that corresponds to the precision prevention assumption. We need additional assessments just because the pathways are multiple, and this dilutes the promises of primary prevention, including in terms of cost effectiveness. Further, well, epidemiology has still a lot to say, classical epidemiology. For example, the prevention paradox put forward by Jeffrey Rose. Well, I will not go into details, but we know that a large number of people at a small risk may give rise to more cases of disease than the small number who are at high risk. Or in other words, a preventive measure which brings much benefit to the population offers little to each participating individual. And this is definitely a conundrum of precision prevention, which leads to the fourth word of caution, because without careful consideration given to equitable access, more sophisticated medical or preventive interventions pose a risk of exacerbating social inequalities in health rather than helping to resolve them. And once again, all these problems have been formalized by epidemiology, for example, in the concept of the number needed to screen or to treat, which doesn't depend only on the effectiveness of the preventive action, but also on the frequency of the outcome. And this is still an unresolved problem that precision prevention will not solve easily. Well, I skip, okay. I like to close with this citation from Bob Weinberg, who wrote a very nice paper in Cell a few years ago, So apparently all one needs, so the doctrine goes, are some computational algorithms to distill data into simple, accessible take-home lessons that would involve mechanistic insights. But this is overconfident presumption. And it refers to the emphasis put on big data nowadays and the use of big data for precision medicine and precision prevention. Thank you.